With every trial, every test, we move closer to the rest. A world where pain no longer stays illuminated by brighter days. Waits for a cure. Rise and shine, rise and shine.
Hi, everybody. Thank you. Thank you so much for having me here. Thank you, Richard, for the invitation and the other organizers for the invitation. It is really a joy to be here. I want to thank you all for everything you've shared today. I want to thank you for sitting through another talk where we're going to talk about immunology, which is my favorite subject, and I'm going to try to make your favorite subject too. But I mostly want to thank you for all the work that you've done in the community and all the work that you've done interfacing with us in research. Like none of what we do in Cure Research is possible without all of you. And I'm going to talk a lot about why I'm hopeful for where we are on the biology side, on the medicine side, but a lot of that builds off of the hope that I get from you guys and from everything that you're doing. So mostly want to thank you. So I introduce myself. I'm from UCSF. I work closely with Steve Deeks, who you may know. If he's made some bad jokes, they're not my fault. Yes, he has. I'm a physician. I work with people with HIV at San Francisco General Ward 86 Clinic. But mostly what I do with my time is I'm an immunologist and a researcher in the lab. I'm trying to understand, and our lab is trying to understand, along with many people in the field, how we can get the immune system to do better at fighting HIV in the body. And so I'm going to be talking about this concept of post-intervention control. And so this is a really high-level overview slide of what I'm going to talk to you about. So if you just get this and the rest of it is a little much, this is what I want you to take home. So I'm going to be mostly talking about a study that many of you may be familiar with. It's a combination immunotherapy study where we tested a new strategy for HIV cure. I'll get into the details, but it involved a combination of a therapeutic vaccine to get T cells going and antibodies as well. And we included a treatment interruption. So we were trying to check to see if these interventions affected the way that HIV was controlled after the treatment was stopped. And this was a small study, proof of concept, but it really showed for the first time a really big impact on the number of people who were controlling HIV to lower levels after stopping therapy. That's one of the big reasons why I'm hopeful. So 7 of 10 participants had better control of HIV after stopping therapy. And the other thing that gives me hope is we actually found what in the immune system helped, what actually seemed to work in those individuals who were controlling. And those are a type of T cell called CD8 T cells. I've been studying them since I was a graduate student in 2004, so I really love them. And then you'll hear, but you do too, and you will too if you don't yet. But you'll hear about how these CD8 T cells pounce on the virus, and that capacity of them to do that is what we think is really important, and what we think we can harness in cure strategies going forward. So we think this matters because we think it shows progress, but also because we think that we can move forward with it. So this has been introduced, you all know it, but antiretroviral therapy, of course, prevents active HIV replication, but is not curative. And that's why we're doing this cure work. And you heard from Nicola, and you all know many kind of aspects of where the virus persists in the body, especially in tissues. And that's why lifelong therapy is required to really keep it at bay in most people. But in this particular moment, it's really interesting and exciting for me as I've been in the field, I guess now 10 years, so I can't say I'm new, but I started my lab 5 years ago, so we're considered still new. But right now in this moment, there really is a global effort driven on multiple different levels by many people to develop all these things everyone is talking about: a safe, scalable, and effective cure for HIV. And of course, you hear it loud and clear, and we all know it. We want that to be eradication of the virus from the body, and hopefully we can get there. But for right now, a lot of what we're seeing movement on is being able to get some kind of sustained control, and that is more feasible, and that's what I'll focus on. But of course, we recognize the bigger goal. So in terms of eradicative cures, I think everyone is familiar, of course, with Timothy Ray Brown's case, and maybe many of the subsequent studies that have been done in individuals who got, stem cell transplants, kind of a replacement of the immune system, and were able to sort of sustain the absence of HIV in the blood in the absence of antiretroviral therapy after getting these transplants. This is not scalable, but this is proof of concept that that can be achieved in the human body. Well, what I'm going to be talking about is a different category of strategies called immunotherapies, where what we're really trying to do is turn this line here of the viral load going up after stopping antiretroviral therapy into one, if we were to take a COVID analogy, we're trying to bend the curve on the virus, right? We're trying to bend the curve. We're trying to reduce the levels of the virus in the body, maybe to undetectable levels if we can, but as best as we can with trying to harness the immune system. And we don't come into this, naively. We know many shots on goal have been, tried, attempted, mostly with therapeutic vaccines, some with antibodies, and many of them, I would say, this is a slide from Steve Deeks. He says they failed. I would say they taught us things, but they didn't achieve this kind of like low level of virus that we were looking for. And so when we talk about trying to achieve that, I think it's important to recognize that everybody has a different curve. Everybody has a different rebound of the virus after stopping antiretroviral therapy. And I really love this. It's called a meta-analysis. It looked at 382 people with HIV from placebo arms in clinical studies coming off antiretroviral therapy. And you can see that these are all individual lines from individual people. People rebounded slightly different. But this is the summary. Of those 380 people, when they came off antiretroviral therapy and they were let, you see where does the virus go? Very few people actually achieve, levels less than 50 copies per mil or undetectable levels. And even if you started ART very early in the course after HIV acquisition, still only about 6%. What about under 1,000 copies per mil, which is low? It's still only about 10 to 15%. So that I want you to remember those numbers, that like 10 to 15%, if you didn't give any intervention, is about where people would wind up with low viral loads. Okay, there are some people who control, but it's only 1 or 2 or so out of 10. So this is our question. How do we move this needle? And so I'm going to take you through 4 short chapters describing where I think we are with this progress. I'm going to introduce you to some of the immune system and some of the players. We're going to talk about what do we mean by HIV control after stopping ART, and then I'll tell you about the combination immunotherapy trial kind of from a high level view. And then we'll spend a little bit of time talking about what I think about is what's next and kind of ideas from talking to others in the field as well. Okay, so there are a lot of players in the immune system. It's very complex. I'm an immunologist and I get confused, so it's totally reasonable. But the general categories of immune cells that respond to viruses, you have like your initial first responders. And so when you hear the term innate immunity, that's that initial response. You have your cell types that are capable of finding those infected cells and killing them. And mostly we think about these killer or cytotoxic CD8 T cells, but these other cells, NK cells that you may hear about, can do that too. And generally speaking, I think we've now talked a couple times about broadly neutralizing antibodies, and we kind of probably know a lot about antibodies from our time with COVID and thinking about vaccines. And so these are made by B cells in our body naturally. And when we give a vaccine, we can induce these antibodies. And where you hear about it in cure research, of course, is that broadly neutralizing antibodies are capable of recognizing lots of different HIV variants. So in cure, the 2 main types of immune responses that we try to intervene on are these T cells, these killer T cells, the cat that I love. Okay, yeah, okay. The killer T cells, and then these antibodies. And you've heard about BNAB infusions, and maybe you've heard about CAR T cells, like kind of giving T cells into the body, or vaccines are another way to kind of stimulate these kind of immune responses. Okay, these are the players. What about the patterns? And how do these players kind of work into those patterns? Well, in our cure studies, when we give immunotherapies and people come off antiretroviral therapy, one thing that could happen is you have a delay in when rebound occurs. And that's kind of interesting because that means you figured out an immune response that was able to do something, right? But it's not the end goal because the rebound occurs again. And that delay doesn't happen very often normally. Normally we see rebound in 1 to 4 to 6 weeks or at max most. So this delay by infusions of antibodies is interesting, but that's not really our goal. It did tell us something that worked. You can imagine a further delay that would be marked by having no rebound, for example, and that's bNAbs can do that. They might induce kind of a better immune response and control. And then another pattern of control is one where, let's say we give an immunotherapy, people come off therapy, and now they're controlling to lower levels. And that is what we say is kind of like a low viral load. That's not undetectable. It might be undetectable, but it's not necessarily, but it's low and meaningfully changed from what, this is kind of what the arrow indicates, is meaningfully changed from where we think people would have been. And so much evidence. We were just talking a little bit, I think Steve Deeks gave a talk on people who spontaneously control infection, right? So we've learned so much in the last 30, 40 really years, right? About people who naturally control. And a lot of that evidence is that these T cells are capable, what seem to be correlating with, you can find these highly functional CD8 T cells in individuals who naturally control. And there's a lot of more accumulating evidence that those T cells might actually be doing something. And so that evidence makes us think that this pattern of low viral control could be mediated by CD8 T cells, but the field hasn't really proven that yet. But that's kind of what the thought has been, was that you potentially could achieve immune control. And then the other part of this, and I'll just go back to this for a second, is that broadly neutralizing antibodies also maybe be able to stimulate an immune response to achieve better control. So I think I had another version of my slides that had 2 other quick slides in it. So I'm just gonna tell you 2 things real quick. So I told you that bNAbs, I told you CD8 T cells we think are probably important for this immune control. I told you bNAbs could potentially do something. I want to just acknowledge there have been, and you guys all are pretty aware, have been a lot of studies of bNAbs. And what we think they have done is sort of made maybe 30% of people do better in control. So I told you the background number is like 5 to 15%. With the bNAbs added, now you get up to about 30%. And one of the most important recent studies that's been reported at conferences, there's some studies on it here, was a study that's out of South Africa, that's out of the FRESH cohort. So young women with HIV who were started on antiretroviral therapy very early in infection, got a combination of bNAbs and a TLR7 agonist. And for the first time in a study in an African country, they underwent a treatment interruption. And there was a lot of work from community members to figure out how to do that ethically, how to do that well. It was kind of an incredible study for a lot of different reasons. But one of the things is they saw that outcome. Now the bNAbs wore off and about 30 or so % of people seem to be having low levels of control. And so they have sort of a platform to be able to study that. So sorry, that was on some other slides. I don't have them here, but I want to make sure I acknowledge that, you know, there are a lot of bNAb studies that have shown a little shift in that needle. Okay, so let me tell you now about our combination immunotherapy trial. So just out of curiosity, how many people here have heard of it? But we did a combination immunotherapy trial, AMFAR trial, the UCSF. Oh, not that many. Oh, great. Okay, cool. So again, high level. So this was an immense study with an immense amount of input and interaction from a lot of different people. So the first, so this is like hearkening on the idea of like, it takes a village. So the first level of the village was Michael Peluso and Steve Deeks led this study. Steve designed it, brought all of these different players together, all these companies, all these collaborators together to say, look, bNAbs aren't doing it alone. Therapeutic vaccines are not doing it alone. We need to put these things together. We need to start combining things. You all need to figure out your legal stuff so that you can all put, you know, a trial together. And we need to figure out, you know, make sure that that's safe, choose the right things and do that. So much work went into designing this study, and putting together the right things. And the two real leaders of the study were Becky, and that's a typo, she's RD, and Megan. Becky Ho runs the SCOPE cohort in San Francisco. It's not Steve, it's Becky. Yes. Yes, and also never like the scientists, doctors. It's never us. It's the nurses. It's the nurses. We know this. So all of this work went into designing this study, and then it's a study that opened in February 2020. So the other slide I'm missing here was a slide where I had, and I'm happy to share this, just an immense amount of work from Danielle, Jeff, Linda D on the CAB, and sociobehavioral researchers who have contributed an immense number of scientific papers to the literature describing how to do these types of studies, trailblazing, how do you talk to your participants about how to enroll, and a lot of the things we talked about today, how do you get inclusive engagement and make sure that you're working with people. But also this opened in February 2020. So how do you deal with the COVID pandemic on top of it? How do you deal with, the ethics of bringing people in for the research study? And so I wanted to really make sure I acknowledge that, because that work was more village required to get this done. And then the other slide I don't have here, I'm so sorry, was a slide of Tom Perot. So he was one of our study participants who, and I would just say, broadly, that the other part of the village are the actual study participants. This was, you'll see, a very intensive study. You can already maybe get that sense. And they put in so much work and so much dedication. And now they also talk about it. And they also come to our lab. They come talk about what the experience is. They talk publicly about it. And that is such an important part. Okay, so what was this say? I already mentioned therapeutic vaccines don't do it alone. BNABs have a little bit of an effect. There was a lot of data accumulating 6-7 years ago that from monkey studies, non-human primate studies, that maybe a combination of antibodies and T cells might do the job because there's some interaction there. So this was really the first time 3 different interventions were tried together in a cure study. It's a phase 1 single arm study. We intended to enroll 20 participants, only could enroll 10 because of the complications with the pandemic. We had skewed a little bit towards people who had started ART early, with the idea that if we saw a signal there, that would give us something to move forward with. If we didn't, then we wouldn't. This was a majority cisgendered male study. We did have one transgender woman. 40% of the participants were Latino. We definitely could do a better job on gender balance here for sure. And then the study was designed to detect a large effect. So this study involved 3 different stages. So I kind of highlighted in red the things to pay attention to. The first stage was, let's get the T cells going with a vaccine regimen. Let's target them against parts of HIV that HIV can't get away from. The second stage was a stage where we gave broadly neutralizing antibodies and this kind of immune activation thing, a TLR9 agonist, with the idea like, let's stir up some trouble under antiretroviral therapy. Let's stir up the immune system, get maybe the virus activated, maybe reduce the reservoir. I will say that stage didn't reduce the reservoir. So that's why we don't think that maybe contributed to the outcomes. The third stage is where we had people off antiretroviral therapy with the antibodies given at the same time. So now you have antibodies slowly coming off. And they took about 15 weeks before people started to rebound. But the key thing was that once they rebounded, we had relatively open criteria to let people, if they went up high and got high quick, right back on antiretroviral therapy. But if they went and stayed low at low viral loads, they were allowed to stay off viral loads. Not every study needs a longer ATI. But when you're doing immune-based things like this and you're trying to say, do you affect that natural curve? It's very helpful to have a little bit more time to say, what does the curve look like? And this study changed the curve. So I mentioned at the beginning that most people with HIV experience viral rebound that occurs very quickly, goes up really high. Viral loads go back to about where you were prior to starting antiretroviral therapy. And that happened in 3 of the 10 participants. But what happened in 7 is quite different. So 2 of the 7 actually had what we call a very slow creep up of the viral loads. Took several months to get even above 1,000 copies per mil. Quite different from what we've seen before. We had 4 people who also kind of slowly went up and then sort of stopped around 1,000 or oscillated, kind of went up and down a little bit around 1,000, several months off antiretroviral therapy. This was the pandemic, so our participants restarted art because they wanted to and because they needed COVID vaccines and all that stuff was happening, right? So, but they didn't meet our restart criteria. They could have gone longer, potentially, but it looked like they were pretty stably controlling despite the fact that the antibodies had waned. And one participant, really remarkably, didn't rebound for over a year and a half. So this is one of the main reasons why I have hope right now. The combination therapy trial moved the needle. More people had lower viral control at lower levels than expected. Now, why was that? And I'll just briefly run through the things that it wasn't. So, was it that the BNAbs were sticking around? No. Was it that we had reduced the reservoir? I kind of already told you we didn't really do that with our interventions. And the cohort generally had smaller amount of HIV in the body because they had started ART early. And this person who didn't rebound actually had a very, very, very small reservoir that actually we couldn't really induce to get out and come out. And so it might have been a very special case. That being said, individuals like that have been studied, taken off ART, and they still rebound. So we wonder whether still the intervention did something there, but we can't rule out the fact that just this was someone who wasn't going to rebound. And then the vaccines, I mentioned T cells. It's great. So we have all these assays. We ran them all. I have a lab of like 12 people. They were like, yeah, we're going to figure out what's going on. We saw no correlation in the lab when we looked at what the vaccine did and the outcomes. Prior to taking people off ART, we were like, gosh, we have to find a correlate here. What's going on? Why can't we find the correlate? And then we remembered the literature. And the literature tells us that these CD8 T cells are probably targeting the virus as it comes out. So then we said, we have to study that moment. We have to study when the virus is coming out. That's our moment. That's the key time we have to catch these T cells. And this is what we saw. We saw that the individuals who had wound up having lower set points, okay, on the left-hand side here on the bottom part. So these people here this is a measure that my very first graduate student, she graduated from the lab, found that they had a huge proliferation of their CD8 T cells when the virus was coming out. So this is telling you that 20% of the CD8 T cells are like out there, fighting, ready to go. Just as the virus was coming up, before the viral load split, these people who went this way had this pouncing CD8 T cell response. We like the pounce, right? Yeah, the pounce on that viral load, right? Whereas the people who, they were coming out and the virus is going up so fast, like we know it does, their CD8 T cells weren't responding at all. They did eventually, but the cat's the pouncer. The cat is pouncing on that viral load and capturing it and I'm not going to go further with that analogy. It's not like complete, right? But it's pretty good. It's captured. Okay, so what did we learn about the T cells here? So I didn't give you this background, but normally people with HIV, because their T cells experienced a lot of virus for a long time before starting ART, the T cells are what we call exhausted. So the analogy here is they're not ready to pounce, and that's the cat just sitting back, relaxing, taking a nap, doesn't really realize there's a little, like something coming out that it needs to pounce on. But when you have the CD8 T cells that can pounce, they proliferate, they start expressing a factor that's called TCF1, that's like my true love, don't tell my wife. So she kind of knows that. My 5-year-old daughter can draw TCF1. So the TCF1, that's what gives them this capacity to pounce. And so when you hear people talk about stem-like T cells at this meeting, and you hear them talk about T cells that have this capacity, it's this capacity to pounce. It's driven by this factor called TCF1, and it allows them to turn into the killer cells that express granzymes. So the question is, how did we turn the T cells into these T cells that were able to pounce? And I'll just say that we're still working a lot of this out. We have to do a lot of kind of deep, fun, interesting immunology, but could this be directly because the vaccine did something? Our assays are not great. The ways that we look at T cells aren't great. So we're applying new technologies to look at the T cells and get a better look at that to answer that question. Or is it something about the BNAbs that either slowed the rebound or caused the T cells to get more activated? And we have a poster here and a short talk given by one of my postdocs at Croy, who talks a little bit about some evidence for that. But I would say the jury's out. What you should take home for this is that the T cells can pounce, and we're still working on figuring out how and why the interventions did that. So in just a couple minutes, I'll just briefly touch on what's next in cure from our perspective. So I think one really important thing is that we recognize that this isn't the end, right? And immunotherapy has to, at a minimum, allow for a plasma RNA level below at which transmission occurs. That has to be useful for people who aren't able to take ART. And transmission threshold, there's some debate, but under 1,000 is probably pretty good. If you can bend that curve on enough people, you probably can reduce transmission levels. But obviously, our goal is to do better. And so how do you know if the cure is working when you have people out getting a therapy? There's a lot of development towards affordable rapid at-home diagnostics, which is kind of what you need, right? If you're like, hey, you're going to get some kind of immunotherapy, you're going to come off your ART, you need to know what your viral load is. And so this kind of work goes hand in hand with what we're doing. So we recognize the combination approach. This was very complex in its current form. It's not effective enough, and it's obviously not scalable. That's not what we were trying to do. We were just trying to prove that we can get the immune system to do better. And now the key hope is that we have something that we think is an actionable correlate. And so as we think about this better understanding of what the immune system can do, we're working towards developing better vaccines that don't require so many doses, that are safer, that are more scalable. That's now enabled by what we learned during the COVID pandemic. Obviously, challenges to vaccine development right now, more broadly, but scientifically, this is a very exciting time to be working on these types of vaccines. And then there's also, and I'm happy to talk more about this, we're working on some ways to potentially engineer T cells that won't require you to take the T cells out, engineer them, put them back in, but are more like the idea of giving a single shot where you can turn some of your T cells into the right ones to be able to fight the HIV. We are a ways off from that, and there's a lot of discussion to be had about it. But again, the technology for that, because of cancer research, because of autoimmunity research, is in such a different place now than it was even 3 years ago. So that's more hope that I have. So just final thoughts. In addition to immunotherapy, I think, we recognize immunotherapy might cause this better control. Maybe we can get full control. But I just want to harken back to the idea that perhaps we do need a combination with something to target the reservoir. And this is a slide from Steve, and Steve has been doing a lot of work in kind of the gene therapy kind of field, thinking about how do we target those reservoirs better. Things we learned from Timothy Ray Brown, things we learned from other people who got stem cell transplants about the ways that might work better. So I'll just end by thanking you all for listening. I'm so grateful to our study participants, our clinical teams, our CAB, and we had 40 authors on our paper and like 100 people in the acknowledgements. So many people, obviously Steve, Michael, Demi from my lab, and Amelia, my whole lab, who keep us all honest. And I encourage to attend CAB events for exactly the reasons people talked about. Like, they need to know what's going on and what people are thinking and prioritizing. And of course, our funders. So thank you. Who is the cat? Great question. It's a Google cat. It's not an AI cat. Let me tell you, we tried to make, we like explored the AI world. You don't go there. My cat is the cutest cat ever and has the most robust pounce, I think. But he like cannot sit still for a picture. I can't, you don't want to see my photos. So he's the spirit of my cat. Yeah.
Rachel? Any questions?
There are definitely questions.
I can.
Hey, Rachel, that's I mean, an amazing talk. So you get the 99 of 100 from me. So that's
Thanks, Linos.
really a great talk. But I have a question that is something I want to ask the CAP here, and also answered by one of my patients very recently, and that is linking also to your last slide. And that's basically, my patients have, or my people living with HIV, have a problem with research that they would engage with if it's supported by the case group. And that is because of the fact that name has been blurred over the last weeks. Is that something you have been thinking about, or is that something that I should not ask you because it's too sensitive?
No, I mean, I think oh yes, sorry, you're right, that's right. The question is whether, from Linos, is whether in this political moment, we're concerned about funding coming from the Gates Foundation. Yeah.
Because of the
relationship between Gates and Epstein. Yeah. Yeah. I mean, yes, of course, these are discussions we all should be having and we should all be thinking about. And I think sources of money are always complex. I know people at the foundation are furious.
Because we are all in funding cuts, we need that money.
Yeah.
But on the other hand, you don't want to have your research where you work the highest standards of GDPR, patient safety to be supported by the whole team. Right?
Yeah, I think it's an important question, and I'd be curious what the CAB thinks too. I think we have small grants from the Gates Foundation, but I think they're doing much bigger efforts. And I was just there a couple of weeks ago at the CAVD meeting, and I think there is a lot of anger broadly in the field from researchers and from foundation members over this. So I think it's definitely a conversation we need to have. I think similar things have come up with other funders as well. So I appreciate the question. I don't have a great answer, but open to thoughts.
Make it the Melinda Gates Foundation.
Huh?
Make it the Melinda Gates Foundation.
Make it the Melinda Gates Foundation. I mean, I wouldn't be opposed.
Yeah, quite questions in the chat, so we're going to take one from the chat and then come back to those.
Okay. Not a scientist, so hopefully I pronounced this correctly. Have you looked at cholinergic signaling in TCF1?
Yeah. Thanks. I see Richard said, I hope Rachel can answer that question. Yeah, we have not specific. So there are a lot of, so I talked about TCF1 because that's a very interesting driver of these like cat-like T cells, but there are a lot of other things that drive that capacity in T cells. And I think this is a question a little bit about metabolism and cellular metabolism and how that contributes. We have a little bit, but not maybe exactly at TCF1 positive cells. We have not applied some of like the advanced technologies to look at that specific question. It is really important to make the general point that TCF1 might not be the thing that you want to exactly overexpress to make the cells better. We need to kind of think more holistically about what's going on and why they're so good.
Thanks. For me, it's just there's a lot of progress that's been done in terms of research, new development, innovative development. But what have we learned from the COVID as we will apply into HIV studies? And Secondly, I don't believe that researchers have done enough in terms of research. But that's the perspective of the South African activist. You know, there's researchers, they have never done so much in terms of the vaccine research. Because we compare the vaccine of COVID, there were lots of acceleration, and there were lots of stages that were skipped, as if it is more of the drastic measure. But we are having a very negative feeling that you never took a very serious step in terms of vaccine initiation or innovation. What will be your response on that?
Those are two great questions. So the first question is, what did we learn for HIV from COVID? And we've learned a ton. I think one of the biggest things we learned actually is how to use mRNA vaccines. And actually that we knew on small scale from smaller scale studies in cancer. And there were starting to be some studies in HIV that was starting to be planned prior to the COVID pandemic that the resources got shifted over to making the COVID vaccines. But we learned that they're safe, and we learned how to scale them. And there right now is all this cool research going into, how to do it so you don't need a cold chain, and how to do it so that they're more immunogenic, and how to get better T cells, and how to make it more scalable for mRNA vaccines specifically. So I think actually that's the biggest thing to me that we learned scientifically. I think there's a lot that we learned socially about infectious disease messaging and public health and all that stuff as well, that has helped us and informed how we do HIV research generally. The second question is this: I agree with you, the appearance that the COVID came around and suddenly there was all this vaccine development. Why wasn't there, or the impression that why wasn't that there for HIV? So first of all, I would say everything that happened with HIV vaccine development and antibody development, everything that happened for COVID came from HIV. The reason that it was able to accelerate was because of that. We were primed, we were ready. We had all those technologies that had been tested. So, but HIV is a much harder virus than SARS-CoV-2.
Okay, follow-up question. If you are reaching on the level of the time.
Yeah.
Duration, that's it. In terms of the time on the duration, on the COVID vaccine,
Yeah.
took a very shortest time. That's everyone was not expecting it.
Yeah.
But in terms of the HIV, it's more than 20 years.
Right. So I would argue, I wasn't there, but I would say that 20, 30 years ago, we thought, oh, we know how to make vaccines. We can just make a vaccine against HIV and this will work. And when that failed, we realized, wow, this is such more complicated infection. We have the reservoir to deal with. We have all this immune dysfunction to deal with. And now we're really, partly because of cancer immunotherapy, learning how to deal with some of that stuff, how to get into tissues, how to deal with things that change a lot. So we're ready to apply that to better HIV vaccines. And I would say the HIV vaccine world has a lot of promising stuff going on right now. That's, phase 1 clinical trials. But it has just been a harder problem. I have to go. Yeah.
No, I'm not pushing you.
Okay.
I just wanted to say a little bit. Can I use it?
Yeah. Sorry. It's like, I'm just talking. Sorry.
No,
So much
you're fine. So that's a very good question. And again, my name is Louis Shackelford, he, him pronouns. I'm the Director of External Relations at the HIV Vaccine Trials Network. And so we have quite an extensive bit of work that happens in South Africa. And actually our lead scientist in South Africa is Dr. Glenda Gray. So Dr. Glenda Gray leads our vaccines initiative in South Africa. Now, recently I'm going to try to pick my words wisely. With a lot of the political shifts that we've seen in the past year, there has been a halt in a lot of the work that we were doing in South Africa. And if you want to know why, I can get into that offline. But the HIV Vaccine Trials Network still exists, and we still have intentions on continuing our portfolio of vaccine work in South Africa. And as Rachel said, a lot of that work right now is phase 1 studies that look at a lot of immune responses that are similar to what Rachel presented. The other thing I wanted to mention is that we cannot take for granted how much resources were poured into COVID all at one time, all of a sudden. And so we have to be honest with ourselves in that we don't see the same amount of resources poured into HIV at one time. And so that's one thing we have to recognize, is that yes, COVID is a very different virus. COVID is a much more simple virus in terms of how it operates than HIV as Rachel said, HIV is far more complex than COVID as a virus. But the other social things around COVID were the amount of resources that we poured into COVID all at one time. And then the fact that just about every scientist, particularly every vaccinologist and immunologist, stopped what they were doing everywhere in the world and started working on COVID. And so we've never seen that type of collaboration happen all at one time around HIV, both in the amount of resources that were poured into COVID. We've never seen that in HIV all at one time. And we've never seen the scientific community galvanize around one specific thing all at one time in that way. So I hope that helps to clarify. And if you want to know more about what the HVTN does in Africa, please shoot me out.
For the sake of time, we'll do one more from the chat.
Great.
Is there any applicability for autologous stem cell transplant as part of the combination therapy?
Is there applicability for an autologous stem cell transplant as part of a combination therapy for HIV? I think we've learned a lot of biology from the stem cell transplants. I don't think it's a scalable therapy in and of itself because it requires a lot of immunosuppression and then a big procedure that has to be closely monitored after you get the transplant. So that's why it's been done in people with HIV who have another indication for it. So I don't think a stem cell transplant is going to be a part of a combination immunotherapy. Do I think that there are other ways to target the reservoir in part that try to excise the virus? Yes. And I think that those strategies are really interesting and there's a lot of preclinical development going on. They're just not there yet for showing clinical efficacy. Thank you all. Thanks for your time.
Quest for a cure.